Good morning and welcome. I'm glad to see such a good turnout here in spite of the recent weather. I'm Bruce McEwen. I'm a professor here and also a faculty member involved in the Science Outreach Program. I want to tell you a little bit about these holiday lectures. They were founded by Alfred Mirsky, who was a professor here for many years, and modeled after the Christmas lectures of the Royal Institution in London that were established in 1825. The most famous of these lectures was Sir Michael Faraday's A Chemical History of the Candle, which some of you may know as a book that was published, and I know I read it when I was growing up in science. The Rockefeller lectures, which are now endowed by a gift from the late Alfred Mirsky, are given every year between Christmas and New Year by a Rockefeller scientist. Like the original Christmas lectures in London, the purpose of these lectures is to reach out to young people to present exciting science. Our holiday lectures are now part of the Science Outreach Program, established in 1992, formerly headed by Dr. Bonnie Kaiser, whom many of you perhaps knew, and headed now by Dr. Ted Scoville, whom I'd like to introduce briefly to you, perhaps to say a few words if you'd like. Just you want to maybe talk about just.
Good morning. I'll make this brief because I know you're not here to listen to me, but I love that we have all these science diehards who fought through the snow to get here this morning and on your vacation to decide to come to this lecture. Over the summer, we have a science outreach program for high school students and high school teachers where you come and do real research in labs. There's actually two professors here. There's opportunities to work in their labs. And I have some information. There's a website which gives you all the details. For high school students, the applications, though, are due January 15th. So it's an amazing way to spend the summer. You learn an incredible amount. And every kid who came last year wanted to come back for a second summer because it's also really fun. We do things at night and that sort of thing. So if you're at all interested in doing research over the summer at Rockefeller University, come see me. I'll be at lunch. I'll hand you one of these. You can go to the website. Thank you all for coming.
Thank you. So, this year's lecturer, Dr. Sohail Tavazoie, joined Rockefeller as an assistant professor in 2009 after 4 years as a research fellow across the street at Memorial Sloan Kettering Cancer Center. Before that, he received his undergraduate degree from University of California at Berkeley, his Ph.D. from Harvard, his M.D. from Harvard Medical School, and the Harvard-MIT Division of Health Sciences and Technology. He completed residency and internship at Brigham and Women's Hospital and Harvard Medical School. And as you will shortly hear, the goal of his laboratory is to generate predictors of cancer outcomes that will guide clinical decision-making and identify and therapeutically target critical nodes required for cancer progression and resistance to chemotherapy. So without further ado, please.
Okay, thank you very much, Bruce. It's really a pleasure to be here, and I know you guys had to really well, actually, we have the president of the university here, which right in the back right there. So he's also here. So it's pretty exciting for me to be able to give this lecture to you today. So what I'm going to tell you today is, actually, let's see what happened here. This will work. A little bit of computer jitters. I'm going to talk to you today about cancer and also cancer metastasis. But let's start this off with a little animation to wake you guys up. So what this animation is trying to show is how cancer forms. It initiates when an individual's normal cells in your body accumulate genetic mutations, allows them to start expanding in those tissues, and allows a mass to form. That's called a tumor. It's not cancer yet. It's called benign because it can't kill the patient yet. These cells can actually generate blood vessels. They can recruit blood vessels. And subsequently, upon recruiting blood vessels, one or a few of these cells will actually break out of that mass and get into the bloodstream. See this clever guy right here? These are your red blood cells. And metastasize. What's metastasis? Well, it's a spread of cancer from the primary tumor to another organ. And you can see it spreads out and starts growing there. That can be your brain, the liver, the lung, etc. So that's just the schematic. So we got a lot of things to talk about today. It's going to be a lot of fun, okay? So we're going to talk about, the first part is really going to be focused on cancer. We're going to arrive at a definition of cancer. We're going to talk a bit about the epidemiology of cancer. We're going to talk about different cancer types. Before we learn about cancer, we need to learn about traits of normal cells, okay? Because cancer arises from normal cells. After we talk about the traits of normal cells, we're going to talk about how the cancers differ from normal cells and what they do to do all the crazy things they do. So what is the molecular basis of that? We're going to talk about good genes, and we're going to talk about bad genes, okay? The good genes are called, anyone know what the good genes are called? Good genes are called tumor suppressors, and the bad genes are called oncogenes. All right. So cancer. Let's just start very simply. Let's sort of get a group-wide definition of cancer. So what is cancer? So I'm going to open it up to the audience. Does anybody, so you guys think about it for a second, okay? Think about what you think the definition of, what is cancer to you? And then once you're confident, raise your hand, and then tell me what you think cancer is. So let's see some hands. All right. Let's see. I think this gentleman here was the first to raise, right here. So what's your name? Soham, and where are you from? Herrick's High School, okay. Why don't you tell me what you think cancer is?
Well, it's an uncontrolled cell growth that results from a genetic mutation in the cell.
That sounds like a pretty good definition. I like that. So there's one additional aspect of cancer, and that relates to what we call malignancy. Anyone know? What distinguishes, so he basically said that it's uncontrolled growth resulting from mutations in normal cells, okay? Now, there's one additional aspect of cancer. Anyone know what that is? Yes, you in green. What's your name? Joel. Joel? From? Oz High School.
It's also harmful.
Okay, it's harmful. What do you mean by harmful?
You can have a tumor that will never really cause a threat.
That's exactly right. So what Joel said was that it has to be harmful. Okay, so he distinguished cancer from a tumor, which is something that can expand, have uncontrolled growth, but doesn't necessarily cause harm to the individual. Harm can be a number of things, right? It can be this cancer spreading to an organ and destroying the organ. It can be a mass that locally invades and destroys structures. Let's say your ability to breathe might be compromised, as well as other mechanisms. So that's, you guys are, why are you guys even here? So let's talk about it. So cancer is a loss of normal growth control. So normal cells divide at a certain rate, and that rate is controlled. At some point, a cell acquires a genetic mutation. When normal cells acquire genetic mutations, they undergo cell death, or called cell suicide. The biologists call it apoptosis. Suicide sounds more exciting. So those are normal cells. How about cancer? Well, in cancer arises when normal cells in our body acquire a series of mutations. So after 1, 2, 3, 4 mutations here, Each of these mutations can confer some advantage to these cells. This allows this cell to behave abnormally and grow uncontrollably, and when that mass has the capacity to kill the individual, that is cancer, okay? So our unified definition is a cell or collection of cells that display unrestrained growth and can kill the organism. So a couple questions. So why do we study cancer? Well, firstly, some of us find it biologically fascinating. I would be one of them. It is also a major biomedical problem, as you all know. And also, understanding the molecular dynamics of cancer can give us insights to how our bodies actually work, how do normal cells behave in contrast to these abnormal cancer cells. So I want to impress upon you the importance of the biomedical problem of cancer. So this is basically a chart showing the cancer death rates in blue within the United States. In blue, we have death rates from 1950, okay, these blue bars, per 100,000 individuals. In red, we have, or dark brown, whatever that is, we have death rates in 2003, okay? And what I want you to see is that what we have listed are some of the major causes of death, okay? And so you can see that in the 1950s and still today, heart disease is a major cause of death. But you can see in the 1950s, it was really outnumbered these other causes as the primary killer. Cerebrovascular disease or stroke also killed a lot of people in the 1950s, still does. But what you can see is that between 1950 and 2003 and even today, we've made dramatic improvements in reducing death rates from heart disease, right? And a lot of that is due to prevention. And same thing when it comes to stroke and cerebrovascular disease, okay? And also you can see from infectious disease and things such as pneumonia, we've made tremendous progress. And the majority of this progress had to do with we had to understand these processes. We had to understand what a heart attack is to be able to prevent it. We have to understand what is the basis of stroke before being able to impact it. We have to understand infectious disease and the causative agents that are responsible for pneumonia and influenza before we could make these impacts. And so the basic science is really key to all of this. But when I was in my training and I was studying, doing my graduate work and whatnot, I was really impressed by this, which is you can see that when you've made tremendous progress in these other diseases, death rates from cancer had not decreased that much, okay? Now this is a sum of, this color is a sum of reductions in certain cancers, death rates, but also increases in other cancers' death rates. But the balance shows that we haven't done as well as we have in these other diseases. So it's a major biomedical problem, and you can see that as time goes forward, really cancer is really the top one here. So let's talk about different cancer types. So you guys have heard about different cancers, and you've heard of people who've had cancers. You might have relatives and family members who've been impacted by this cancer. The one thing that we like to do is in order to understand processes, we'd like to sometimes, when we can, when it's appropriate, to separate them and categorize them. So when we think about cancer, we as the cancer biologists and oncologists think of different types of cancer, different broad categories. So one of the categories we have are called solid cancers, okay, solid. And these generally arise from epithelial tissues, such as the breast, the prostate, the colon, skin, okay? So this can be breast cancer, prostate cancer, colorectal cancer, skin cancers, various skin cancers such as melanoma. And these cancers kill the organism by breaking the basement membrane, okay? In contrast to that, we have liquid cancers, okay? So solid cancers, liquid cancers, pretty easy. And these, example of this are leukemias, okay? So how do leukemias kill individuals? Well, the main way they kill patients is by expanding and preventing the normal expansion or function of other cell types, okay? So what leukemia cells can do is they can expand in your bone marrow, okay? And they can compete for the space that other progenitors for red blood cells, white blood cells, or platelets, they can compete against them. And then that can cause these other cell types to be at a disadvantage. So now I have questions for you guys. So based on that understanding, let's see if you guys can be an oncologist, okay? Or let's see if you can be doctors and see if you can predict what the symptoms are of a person that comes into the emergency room who might have leukemia, okay? So now, so let's see if you guys can predict that. To do that, you have to think about what happens if the person's red blood cells are competed out, okay? So you guys can raise your hands. So someone tell me, this gentleman up front, what's your name? Lee? Yeah. Okay, Lee. Tell me, what would be the symptom of someone who has leukemia? One symptom.
Possibly shortness of breath.
Wow, pretty impressive.
The hemoglobin.
Yeah, so shortness of breath due to a reduced hemoglobin. Okay, that's exactly right. So what would happen to your oxygen levels in that person? Lee? They would decrease. What is that called? Does anybody know what it's called when your hemoglobin is low or your red blood count is low? Anemia, exactly, okay? So one symptom of, yeah, do you have something to add? Okay, yeah, you got the next one. So basically, you guys nailed it. One symptom of someone presenting with leukemia is anemia, shortness of breath, low hemoglobin, low oxygenation. All right? Pretty impressive. All right, now let's go to the second one. What happens when your white blood cells are reduced? What's your name? Hannah. Okay, Hannah, tell us, what happens what is another symptom of leukemia?
I know another one is like bruising.
Bruising?
I'm not sure why.
Okay. We'll come back to you because that's the third one. So, so who's got, let's see, someone new. Yes, you. What's your name? Ashley. So what is a symptom of leukemia arising from a reduction in white blood cells? Weak immune system. So what happens when you have a weak immune system? Anyone? Yes. What's your name? Leora? Yes. What happens when you have a weakened immune system?
Your body's not strong enough to fight off.
Fight off what?
That harmful things that you're not strong. So
Okay. Harmful things. What are those things? Yes. What's your name? Tiana. Infections, exactly. So the individuals present with infections, okay? And so an infection. And what happened last time you guys had an infection? What happened to your temperature? Went up, right? So fever, infection. Excellent. All right, so now, what about platelets? And we'll come back to you. You said, what do platelets do? They clot, okay? So she said that you get bruising. So that's one of the symptoms. That can happen. What is another symptom when you can't clot? Yes. They won't clot. So what happens? You bleed. So in these individuals, they don't even need a cut. They can actually, their platelets can be so low that they can just have spontaneous bleeds, okay? So you can have bleeding from your gums, You can have bleeding from various other areas within your gastrointestinal tract. So if someone presents with shortness of breath, low oxygenation, low red blood count, a fever or an infection, and bleeding, chances are, it's not certain, but they could have leukemia. There's many other things that they could have, but that's a possibility. So you guys did great. You're all doctors now. Okay, the third category is lymphoma, okay? And that is an expansion of lymphoid cells. Lymphoid cells reside in lymph tissue. So you guys have all had situations where you've had a cold and you have swollen lymph nodes. You have these lymph nodes all over your body. And these are where lymphocytes and other cell types reside. And a lymphoma occurs when those lymph nodes expand. And how do they kill patients? Well, one way that they kill patients is by compressing on vital structures. Okay? So these things can get so big, they can start compressing on certain structures, and they can kill the person. Can you guys give an example of what might happen? Like what kind of structure? Yes. Exactly. So a lymph node compressing your windpipe or your trachea, and that would basically keep you from being able to breathe. Any other ideas? Important vital structures? Lungs, lots of structures. Yeah, lungs, large blood vessels. Okay, so exactly. All right. So let's take a look at the body. So this is sort of anatomy of different organs, and let's go through this and ask the question: where can cancers arise from? Okay. So can cancers arise from the thyroid gland? Yes. Can they arise from the clavicle, bone? Yes. Can they arise from within the spleen? Can they arise in the stomach, the colon, the small bowel, small intestine? Yeah. The liver, the lung, the pleura. So what's the answer to this question? Everywhere. That's exactly right. Okay, so let's talk a little bit about the nomenclature. So medical language can sometimes be a little challenging, and but sometimes you can simplify it, and it's not that bad. So what do they call epithelial cancers from the breast, prostate, lung, and colon? They call them carcinomas. Okay, and then the question is, what's a sarcoma? You guys have heard sometimes the word sarcoma, and that's simply a cancer formed from a cancer of the connective tissue, such as the bone, the cartilage, or muscle. Okay, that's what a sarcoma is. Glioma is a cancer derived from cells within the central nervous system, typically glial cells. That's glioma. Germ cell cancers, so ovarian or testicular cancers, those are called germ cell cancers. And lymphomas and leukemias, which we already talked about, they're derived from cells within the circulation. Okay, so let's talk about a little bit about the incidence of different cancer types. Okay, so by the end of this lecture, you guys will be experts in oncology and basic cancer. Basic biology cancers. So here are the statistics that depict the most common cancers in men and women, okay? And so let's look at the most common one for men. So this is incidence, like how common is it for a given man in the US to get prostate cancer during his lifetime, okay? And so the other way to think about it is, of all the cancers that are formed, okay, what is the prevalence? And what is the most prominent one? Here you can see that prostate is the highest one in men, followed by lung and bronchus cancers, so lung cancers, followed by cancers of the colon and rectum, followed by the bladder, melanoma. Lymphoma is down here, okay, at 4%, and leukemias are down here at 3%. Pancreas is low, and many of the other sites all add up to 19%. For women, it's similar, but a little bit of a different story. The primary cancer that they get is breast cancer, okay? Followed by lung, colon, rectum, uterus. They also obviously have similar profiles of non-Hodgkin's lymphoma and other cancers. Now let's look at it a different way. Let's ask about which are the deadliest cancers, okay? And for men and women, it is lung cancer, okay? And for men, second on the list is prostate, followed by colon, and you can see pancreas has moved up because it's a deadly cancer. And then for women as well, pancreas has moved up, and you start with lung, breast, and colon, okay? So let's now, it's sometimes useful to look at these types of statistics and look at the epidemiology. Before we understand a process, it's important for us to basically have a sense of its prevalence, its dynamics, and try to gather for ourselves a sense of causality. Can we look at the incidence of different cancers? Can we correlate those with different things individuals are doing or different trends in society and try to identify cause and effect? So this is what scientists do in laboratories. But there's a vast amount of data out there. And researchers can basically take advantage of that data, and historically they have, and they've found correlations and associations between certain things and other things, and then they've tested whether those associations in fact are causal, okay? So let's take a look at this chart, and I think if you look, so what does this chart show us? It shows us cancer death rates among men as a function of the year, okay? And so what is something very dramatic that people see here? You can see this here, this incidence of lung and bronchus cancer. Now this is the hardest question I want to ask you guys. What do you think is responsible for this? Smoking. But back here, people were saying it's not smoking. They were saying it's not smoking. It's not. So you're saying smoking is responsible for this. What do you think is responsible for that? Yes. So what's your name? Jared. Jared is saying anti-smoking measures. Cleaner vehicles, etc. Okay, so clearly anti-smoking measures and our realization that this increase had to do with the increase in smoking in the population allowed society to do this. Okay, that's pretty cool, right? I mean, we're way up from where we used to be, but this is a good thing, okay? And what else is interesting is there's also this trend in green. If you look at colon and rectal cancer, what do you guys think this decrease in trend is due to? Anyone? Yes, back there. Colonoscopies, exactly. So that's one thing. It could be other things, but clearly colonoscopies have been shown to reduce, to catch early stage cancers, and reduce the total incidence of cancer progression. Okay? So let's look at women. Okay. So for women, let's look at this. Again, this is lung and bronchus rates. Same thing, right? But you can see it's a little different. It's a little delayed, okay? but we still have to bring it down. Okay? Let's look at this. How about this? Breast. So this is breast cancer. This is going down. What do you guys think this is due to? So self-examinations could help, and also somebody else said it, mammograms. Mammograms, clinical breast exams, and an awareness of the fact that detection of early stage breast cancer can actually improve survival. Great. So let's now, we have sort of broad understanding of cancer incidence and cancer types. Let's talk about features of normal cells within our bodies. What I want to do is we're going to focus on epithelial cells, but many of the things that I want to talk about also apply to lymphoid cells and leukemia cells, okay? The main distinction being that the epithelial cells we're going to talk to, we think about this interaction with the basement membrane, and that's really what constrains these cells. So let's talk about features of normal epithelial cells. So what I've shown you here is a section of basically lung cells. So these are sections of lung, and what you have is these are each cells. You guys see the nuclei, and these are each cells. They're long. They're called columnar cells, and they have these little things on the edge. Who knows what those are? Cilia, right? Okay, great. So let's talk about features of normal epithelial cells. You have specialized cell types, okay? Within a certain organ, you have cells performing certain functions that cells in another organ won't perform. So in this case, these cells have cilia. That allows them to move particulates up and out of the body and allows you to cough and get rid of foreign invaders. Okay? In between these cells, you might have other cell types that can secrete mucus, et cetera. So there's specialization, okay? You also have differentiated cells. Okay? So these cells that I just talked about, they're differentiated. Okay? Just like many of you will go to become famous scientists and doctors and engineers and whatever you wish to go into, you're specialized and you're differentiated. There's polarity, okay? There's a directionality. The cilia are over here, they're not over here, okay? There's polarity. In normal tissues, you have stem cells. Those stem cells are the ones that can generate cells that can basically give rise to offspring. And these cells are typically at the base, connected to the basement membrane, and they make decisions about giving rise to progeny. These cells have constrained multiplication rates, okay? Their division rates and multiplication rates are constrained. These cells are attached to the basement membrane. Not all of them, but as you guys can see, a large number of these cells basically have fine attachments to the basement membrane. Detachment from the basement membrane for cells that are supposed to attach leads to cell death. Okay, the suicide program we talked about. Cells have attachment to their neighbors. Okay, and cells require protein growth factors, certain level of growth factors to stay alive, just like you guys require a certain amount of Dunkin' Donuts and juice, okay, to stay alive. Cells copy their DNA precisely. So they have editors that read their DNA, make sure that everything is, as it's being copied, everything is copied precisely, okay? If they find errors, these errors are called mutations, they try their best to repair those errors. It doesn't always work. So another feature of normal cells are that they can activate cell suicide programs, apoptosis, okay? This is another tissue type. You can see this is the basement membrane. You have the cells here. Clearly you can see the nuclei, but you can see the cells towards the top. Many of them, their nuclei are gone and these cells are dying. So what happens? How is cell death activated? If cells certain cells that detach from the basement membrane, they will undergo cell death program. Cells can undergo cell death programs if they lose attachments from their neighbors. If cells have an internal clock that tells them that you should not multiply more than a certain number of times. Okay? When normal cells multiply more than, when they approach that limit, they undergo cell death. Okay? It's a nice intrinsic mechanism that keeps these cells from becoming cancerous. If cells generate mistakes in their DNA during replication that is not corrected, if they have a mistake that they can't correct, they will activate this program. If cells outgrow the supply of growth factors, so there's a certain limiting amount of growth factors that are present, if cells outgrow that supply, they undergo cell death. And also, if cells don't have sufficient blood flow, they die. So what does this look like? Okay, so these are cells. Each of these is a cell in culture. Look at the arrows. These are 2 cells, and boom. Game over. Okay? They've intrinsically, we're going to repeat this. The program has told them, you guys are out. And it's an active process, guys. It's not just like they just sort of keel over and die. It's an active program that starts out, and it's a molecularly coordinated. And it involves mitochondria and a bunch of proteins that start chewing things up, and it's highly effective and fascinating. Okay, so in summary, normal cells have many safeguards that control, and these control systems guard against cancer formation. Nonetheless, very rarely, okay, we have massive numbers of cells in our body, and over your lifetimes, it's just unimaginable how precise everything is. Nonetheless, rare cells accumulate mutations in genes that code for these surveillance systems, and cancers form. Okay? Now, in certain cases, in many cases, these cancers can actually still be detected by the immune system, which eliminates them because they don't look right. Okay? But in other cases, these cancer cells are quite clever, and they've gotten rid of stuff on their surfaces that makes them look abnormal, so the immune system can't even see them. They go under the radar, and they escape. So cancer cells are not restricted by rules and surveillance systems of normal cells. Okay, so let me show you an example of unrestricted growth. So we previously showed you normal cells. Now what we have here are some breast cancer cells in culture. Okay, let's see what unrestricted growth looks like. Pretty amazing, right? You can see these guys just, and they'll keep doing this for as long as possible, as long as they have the building blocks. Okay? That's a good question. It's not. It's actually sped up. Yeah. So basically, when that happens, this is what you get. This is an image through an endoscope in the colon. And you can see this colon cancer protruding out. Okay, so cancer cells will multiply indefinitely as long as enough building blocks are available. They have lower growth factor requirements. They just don't care. They're like camels when it comes to water. They can just keep going. They can survive and divide without attachment to the basement membrane. They don't require that ground to stand on. They can survive without attachment to neighboring cells. They don't differentiate. Okay, that's really important. So, as I told you, normal cells differentiate, cancer cells don't. They've turned off the intrinsic suicide machinery. Okay? And they don't repair their mistakes in their DNA. And many of the genes that correct those mistakes are eliminated. So they keep accumulating further errors at a certain rate. And by doing that, they can start doing things that normal cells can't. And they're also not limited by the inherent blood flow of the system. They're really clever. They say, why should we be limited by what's out there? Let's bring the blood vessels to us. Okay? How do they do all this? So in normal cells, safeguard activities are accomplished by proteins called tumor suppressors. Okay, tumor suppressor genes. And these tumor suppressor genes, these are the good guys, right? These are the good genes. They're inactivated in cancer through mutation. And to become a true cancer cell, a normal cell must develop mutations in multiple tumor suppressors. So that's another safeguard we have. We don't just leave it to one tumor suppressor in our cells. We've got many. Okay? And for cancer to occur, multiple tumor suppressors have to be taken out. It's like having many security guards. So tumor suppressors, how do they work? Are they in isolation? Well, no, they work in pathways. So you guys have probably learned about signaling pathways in your course, but we're going to have a little game here, a little demonstration involving people. We're going to try to give you the concept of signaling pathways. And so I need, let's see, I need 1, 2, 3, 4, 5, 6, 7, 8, 9 people. Oh, lots of volunteers. All right. You're going to come here. You're going to stand here. You're the receptor. Exactly. So your arms are going to go up. All right. Excellent. So you just stand there. That's right. All right. So he's a receptor. So he's the epidermal growth factor receptor, okay? And what I have here is I have epidermal growth factor. It's actually chocolate. In the form of chocolate, okay? You are the gene called PI3 kinase. You're right next to him. So stand here, all right? And so you can actually just touch his shoulder. There you go. He won't bite. All right. You're HAT, another gene. All right. You put your hand on her shoulder. All right. You're mTOR. You put your hand on his shoulder. All right. Now the rest of you. All right. I need a tumor suppressor. All right. Let's see. You're going to be the tumor suppressor. See. You basically block. Where's PI3 kinase? That's you. All right. So you put your hand there. So you're going to prevent her from doing what she's going to do. All right. You'll see. All right. Now the rest of you are. You're basically. All right. You're a transcription factor. Okay. So you'll also put your hand right there. You're in the nucleus. Okay. And then you are a gene that the transcription factor makes. So you'll sit in the back. When she's ready, she'll let you go. All right. The rest of you, you're all going to come together into a circle. You guys are the cell. All right. 3, 4, 5. We need one more. All right. You come down. All right. We're almost there. We're almost there. All right. So. All right, you guys are the cell, okay? So what we're going to do is, this is the signaling pathway, and the signaling pathway has to transmit information, right? And in cells, typically, one form by which the information is transmitted is by phosphorylation, right? Phosphorylation. So the phosphorylation is going to be this laser pointer that they're going to hand from each other. So the phosphorylation goes on each protein, and it gets transmitted down. When it gets transmitted, it activates that gene. Okay? So you are the first gene. So let's hear. You're going to have it first. You're going to hand it to her. Okay. She's going to hand it to him, and so on. Okay? Once it's handed to once it's handed to you, you turn on the gene goes over to the cell, and it splits the cell in two. Okay? It divides those guys into two cells. Okay? And so you just hang out there. So you can hold your hand here. All right, here we go. You guys ready? Before you do that, you need the growth factor. When I give you the growth factor, you hand off the phosphorylation to the next gene, and you phosphorylate her, okay?
So I have to eat the chocolate?
You can eat the chocolate. You don't have to, but here we go. Growth factor. There you go. There you go. There's the phosphorylation. It's going down from one gene to the next. They're being phosphorylated. Transcription factor gets activated. The gene goes, and the gene splits the cell up. Nice. All right. Excellent. Excellent. All right. Let's do it again. So now we have, let's do it again. The cell gets back together. We're going to do it again. All right. One more time. There's your phosphorylation. All right. Ready? There's the growth factor. There you go. These proteins are being phosphorylated. The gene is made, splits up the cell. Excellent. That's when the cell has permission to divide because there's a growth factor telling it to divide. Now what happens? Typically, we have tumor suppressors, okay? So what do? Okay, so here's what happens. When a tumor suppressor is activated, even if we give the growth factor, the tumor suppressor puts a block. Where's it? Sorry, here. You're going to take the laser pointer. Okay? Here we go. This is your laser pointer, okay? You're going to take it from her. So wait, hold on. You're going to give it to her, okay? But look, yeah, you take it. Now the tumor suppressor grabs it and takes it off. What happened? All right? So what you have is you have no more cell division. They're stuck here. Got it? So this is the tumor suppressor. It prevented the phosphorylation of subsequent proteins, and it prevented the gene from being transcribed, prevented the gene from separating the cell into subsequent cells. Okay? All right. Excellent. Give a hand to these guys and gals. And everybody gets a growth factor. Those were tumor suppressors. They're the good genes, right? Genes that antagonize the activity of tumor suppressors are called oncogenes. So remember the guys that were handing off the phosphorylation event? Sometimes their numbers can increase. So let's say here, PI3 kinase or AKT, the levels can go up and they can become stronger and more active. Okay, that's what can happen in cancers. So they can actually take that phosphorylation and pass it down to the next gene despite the presence of the tumor suppressor. Okay? So the oncogenes are the bad guys that antagonize the tumor suppressors. And oncogenes are typically activated in cancers by increased expression. Okay? Tumor suppressors are inactivated in cancer by mutations, such as deletions and changes in their amino acids. And cancer cells typically have multiple oncogenes activated, okay? So let's talk a little bit about history. So the next thing we have to think about is how does this all happen, right? Everything happens in the cell through molecular interactions. Okay, these are all molecules inside the cells that do these things. They're the protein effectors and metabolites. And this is really a hard thing to figure out how cancer forms and how it progresses and what underlies it, right? It's just, if you think about it, just seeing a tumor in a person, it can be complex. It can arise from so many different tissues. It's hard to know what's going on in it. It's not simple. It's not a bacteria. Okay, so it's been very difficult to understand the underlying principles that govern the rules of cancer. But really, we really are indebted to this individual, Peyton Rous. And he actually revolutionized cancer biology. He isolated the first tumor-causing virus, and he did it at Rockefeller University. That's right. That's right. He did this in 1911, and he used to be sitting just where you guys are sitting, okay? He used to be on this campus, and he used to do this work right here. And he got the Nobel Prize in 1966, okay? Pretty cool. So another revolutionary biologist was Saburo Hanafusa, and he characterized the first oncogene. And he also did that at Rockefeller University. So one of the things that we should talk about is one of the most important things for us to understand before we start getting a little bit deeper into the biology of cancer is the cell cycle. And I'm sure many of you have learned about this in your biology classes, okay? How many of you have learned about the cell cycle? Raise your hands. Wow. Almost everybody. Fantastic. All right, so let's go over it, okay? So the cell cycle has different phases. Okay, cycle begins here. You have a cell. You have G1 phase where the cell grows. You have a point at which the cell decides whether to go further forward or not. It goes from G1 to S, and in S, you get replication of DNA, right, the synthesis phase. In G2, the cell prepares to divide, and then you have mitosis, and then you have cytokinesis, and you divide the cell in two, okay? Now, this is the principles underlying this cell cycle process are that it is highly ordered and heavily regulated, okay? Because this is what governs whether a cell multiplies or not. So you really need to regulate it. You really need to have checkpoints. You really need to make sure that everything's all lined up and you have enough growth factors and you've got enough nutrients to go through this process, okay? And let's see, here we go. So the guy who was in the back of the room was the guy one of the main individuals who figured this thing out, okay? His name is Sir Paul Nurse. See, I didn't tell you guys he's a sir and a knight. But he is. And he's the president of Rockefeller University. And he basically was responsible for discovering the molecules that regulate the cell cycle progression. He's a jolly man. That's right. Okay, so let's talk about how oncogenes and tumor suppressors do what they do, okay? So now we're getting to a level of understanding that you guys would get either in a college class or maybe even in graduate school. So some of the things we're going to talk about when you're getting a Ph.D. in biology, you would be learning these things. So it's very exciting. Quick, what time is it? Quick question. 11:25. Okay, so we go till 12, right? Excellent. Okay, we're doing well. So you guys have to focus now. We've got about 30 minutes left until pizza time, and so let's talk about this. So as I told you, we have the cell cycle, right? G1S, G2M. Okay? Goes in circles, cells divide. There are two key security checkpoints, right? Just like those security agents at the airport that pat you down and they get paid for that. So basically, we have these two checkpoints, the G1S checkpoint and the G2M checkpoint, okay? And the cell really makes sure everything's in order before it takes a decision to go forward beyond these checkpoints, okay? For the cell to progress from G1 to S, certain proteins need to be activated, just like we talked about, right? These phosphorylation events. And that happens as a result of two two proteins coming together, CDK2, cyclin-dependent kinase. It's a kinase, so it puts phosphate groups on proteins, and cyclin A, a protein that interacts with it but has a very short lifetime. In order for CDK2 to work, it must have its partner with it. Okay? That's required. When its partner comes, joins it, it can go and it can turn on proteins and basically push the cell through the S phase. Same type of thing happens at this other second checkpoint. As cells go from G2 to M, proteins need to be activated. Okay? Here, you have another cyclin, another protein that also has a short half-life that binds this gene, this protein, cyclin-dependent kinase 2, phosphorylates a bunch of proteins, and allows the cell to go through this phase. Okay? First this happens, and then this happens. But what dictates this process? What sets it off? Well, just like anything, one of the main events that puts things into motion is the expression of genes, transcription. That's how messenger RNA is made, and messenger RNA is converted to protein. Those proteins are the effectors in our cells. And this occurs when a transcription factor called E2F binds DNA and transcribes RNA that makes these genes. Okay? Now, how do tumor suppressors work? Tumor suppressors work by binding this transcription factor and preventing it from turning on the gene, turning on these genes. When that happens, you don't have these genes, and you don't have cell cycle progression. Pretty straightforward, right? Excellent. So this is a tumor suppressor. Again, it acts by suppressing this gene, E2F, which drives the cells through the cell cycle. This gene is called retinoblastoma, okay? And it is a tumor suppressor. It's a good gene that's present in our bodies, in all of our cells. And right now, as we speak, it's preventing cells from becoming cancerous in all of our bodies. Okay? So cancer, in the majority of cancers have gotten rid of this good gene. Okay? When they get rid of it, the transcription factor goes, binds, and it activates this process. Okay? So what happens? So we're all lucky, right? We have 2 copies of this gene, retinoblastoma. Okay? But as we age, we might have a mutation in that gene, and that inactivates that tumor suppressor, and cancer forms. Okay? Some people are not as lucky as us, and they actually inherit a mutation in one of their retinoblastoma genes. So they already have a hit. We require 2 hits, because we have 2 copies of it, for cancer to form. So it's a very unlikely event. These individuals have lost one of their retinoblastoma gene copies, and so they're vulnerable. And if they get a hit in that other gene, cancer forms. And this is an example, and this is retinoblastoma. And you typically see it in young children, and it's very sad. And what you see is you actually see kids which basically have this whitening in their. You can see the whiteness through their pupil, and then that's because they have a tumor inside their eye. Okay, that's why it's called retinoblastoma, because it was originally discovered as the gene that is whose loss is responsible for this type of cancer. Okay, so what happens is these kids actually the tumor is removed, and actually you can basically replace the eye and cosmetically, and you can actually today you can actually replace it, and it actually looks. It's very hard to actually tell that someone actually had retinoblastoma, and these individuals much of the time can actually function perfectly normally, and most people can't even tell that they had this. Okay? Now, how else can you get cancer? Well, you can be unlucky and have mutations in cancer, but you can also, infections can cause cancer. Okay? So what is one infection that can cause cancer? Well, there's a virus called HPV, the human papillomavirus. Okay? And does anyone know what cancer human papillomavirus causes? Yeah. Cervical cancer. That is exactly correct. So the question is, how does it do it? It's pretty fascinating. It's pretty amazing, okay? This virus has figured out over its many, many, many years of evolution and contact with its host, it's figured out what our cells are doing and how and which protein is most important, okay, for acting as a guardian of our cells. And it said, I'm going to figure out a way to take this guy out. Okay? How does it do it? It creates a protein, okay, called E7. And E7 binds retinoblastoma and takes it out. And when it does that, the transcription factor binds, activates the genes, cell cycle progresses. And this is a picture of a cervix. Okay, so this is the uterus, and this is the cervical. This is the cervix right here, and you can get cancer on the cervix. Okay, luckily for us, we have very good methods of catching this virus at very early time points, and we can basically, in the United States and in developed worlds, this is really not a major problem. But in other countries outside the US, it is a really, really a major problem. And we have very inexpensive methods to combat this cancer, and hopefully we can continue to spread this, these methods to developing countries so that they are not faced with this problem. Okay? So I've talked to you so far about how genetic inactivation can cause cancer. We've talked about how viruses can trick the system and can cause cancer. And the other thing we want to just talk about is how growth factors can cause cell cycle progression. And in normal cells, growth factors work by inhibiting this protein as well. Okay, they pull it away for a certain period of time. They let the cell cycle go forward, and then when the growth factor has had its activity, then it's blocked, and then the cell cycle stops again. Okay, so now we're going to talk about another gene, and that's called p53. Okay, so this is another good gene. This is called the guardian of the genome, okay? So I told you that in cells, we have many tumor suppressors, okay? And in cancers, many tumor suppressor genes are inactivated. Retinoblastoma is a type of tumor suppressor, and p53 is another type of tumor suppressor. It's pretty famous. It was actually called Molecule of the Year. So in science, it's pretty cool. So we have this, you have the Times, Person of the Year, and in science we have Molecules of the Year. We think that's cooler, and there's a lot of competition. The molecules they have a lot of marketing and advertising. They try to get on Jay Leno and David Letterman and Conan, and they try to promote themselves so they could be Molecule of the Year. But p53 is really an amazing molecule. Okay. So it is actually a transcription factor, okay? So this is the DNA double helix, and this is p53 binding it. And it's an amazing gene because it can do lots and lots of different things, okay? So how does it work? Well, p53 is activated when cells suffer DNA damage, okay? So it can actually sense. Other genes can sense DNA damage, and they tell p53, hey, p53, we've got trouble. Okay? And what does p53 do? p53 can do two things. It can do two different things. It can say, let's stop the cell cycle. Let's assess the damage. Let's fix the damage. And if you can stop the cell cycle, and if the damage is fixed, p53 can then give the green light. Okay? But p53 can also just, if the damage is not repairable, or if p53 is in a bad mood, it'll just cause the cell to die, okay? So you get apoptosis, and you eliminate that cell. Okay? And this leads to genetic stability and cellular stability. Okay? Other things can cause p53 to also be activated, such as cell cycle abnormalities. So during cell cycle, everything has to be done correctly. Chromosomes have to line up correctly. Everything has to be orchestrated in a correct manner. And if there are subtle abnormalities, p53 can be activated, and it'll just take that, it'll stop the cell cycle, try to fix things, and if not, you'll have cell death. So how does p53 work? Well, one of the things p53 does is it prevents this transcription factor from activating the cell cycle, okay? It basically, p53 is a transcription factor, so it makes a gene, it transcribes a gene, and that gene ends up blocking this process. And you get no transcription and no cell cycle progression. And it can also, independently of preventing cell cycle progression, it can actually activate cell death, okay? And again, this is an active process where genes are made. Those genes will basically code for proteins, and those proteins can essentially start chewing up cell contents, and basically the cell can die. So what have we learned thus far? We've learned that cancers are diverse, and they can start in many different tissue types. We've learned that humans primarily suffer from epithelial cancers, okay? So not all organisms suffer from the same types of cancers. We as humans typically primarily get epithelial cancers, such as prostate cancer, lung cancer, colon cancer, breast cancer, etc. While other organisms, such as, you guys know probably dogs and cats, they can actually have a prevalence towards other cancer types, such as sarcomas and certain leukemias that we rarely get. Cancer results from accumulation of genetic lesions, okay? You have to have multiple hits. Tumor suppressors are inactivated in cancer, okay, through multiple mutations. And really this is, so one can say, there are two ways to look at this. One can say, why is it that humans get so much cancer? But if you guys just think about your bodies, think about all the cells you have in your bodies, think about all those cell divisions, think about all the multiplications. You can look at cells under the microscope and just imagine every time these cells are dividing and your genomes are being copied precisely and actively, the real question is, how come we don't get cancer more often? Okay, right? It's an amazing system that keeps cancer at bay. And one of the reasons that I've told you about is there are two reasons. One is that you have multiple genes that safeguard this process. And in order for cancer to form, you have to inactivate multiple genes, and you have to basically get rid of many tumor suppressors, and you have to activate multiple oncogenes. Okay, and these safeguards have to break down, and the immune system has to fail, and the tumor has to get enough blood vessel. So there's a lot of ifs, okay? So tumor suppressors and oncogenes converge on the cell cycle and the apoptosis programs to prevent or promote cancer, respectively. So it's tumor suppressors prevent cancer, and oncogenes promote cancer. So we just talked to you today about two central tumor suppressors, retinoblastoma and p53. And we've also talked about those other genes, those oncogenes that we talked about before. So if you look at this, many of these other genes can actually act as oncogenes, okay? So if you have high levels of these genes that promote the cell cycle, such as the cyclins or the cyclin-dependent kinases, in cancer, you can have high levels of those. So they can basically just promote this cell cycle progression. And this is a good gene, this is a good gene, and you can have increased levels of growth signals, you can increase levels of these other kinases, and so that's basically what we have. So, all right. so it seems you guys are eager for lunch, so just a couple disclaimers. So these are just a few of the. We've just talked about a few of the genes involved in these processes. The mechanisms are vast and complex. There are many, many other genes that also participate in these processes. And most of what we've talked about this morning are basically the mechanisms that initiate the cancers and the early phases of cancer formation. And what we plan to do is in the second half, we're going to start off talking about how cancer progresses, okay? We're going to talk about how cancer goes from a primary tumor site and how it spreads and how it basically leads to the death of the organism. And that process is called metastasis. So that's the first thing we're going to talk about. We're going to talk about the molecular, we're going to talk about the cellular basis of that. We're going to talk about the history of how we came to understand the underlying principles of metastasis. We're going to talk about some examples of molecules involved in that process. And then we want to talk about screening. We're going to talk about how you guys can actually be oncologists today by helping others out there and promoting both a healthy lifestyles that prevent cancer. So lifestyle changes, we know there are a number of lifestyle changes that can reduce the likelihood of cancer, but also so you can actually be advocates for those individuals, probably your parents and grandparents, to make sure that they actually undergo screening processes. And so the best way we can combat cancer that we know of is to prevent cancer. So that's sort of basically the most important thing. So we have 15 minutes, and I'm just trying to, let's see a show of hands. We have two options. One is you guys can ask some questions. The second option is you guys can go and have your pizzas, and some of you who do have questions can come down and ask them from me. So let's take a vote. All right, here's what we have. We'll ask questions, we'll compromise. We'll ask questions for 8 minutes, and then we'll disperse. All right. All right. All right. You guys have microphones on either side of the room. Why don't anybody who has questions walk to those microphones, and we'll start taking those questions. All right, first question. Tell us what your name is and where you're from.
I'm Sean from Center Ridges High School. And I noticed on the graphs about the men and women with different types of cancers.
Yep.
And the stomach cancers, the number significantly decreased over time.
Yep.
Why?
No, I think that's... Let's show you that. So that's a very good question. And so the actual... So did you guys understand this? He's basically really, really great observation. And he's saying that if you look, the rates of stomach cancer have actually decreased here, okay? And both in men and women, okay? And so, and the question is, why has that happened? So they're actually, so this is a really good question. There are a number of different, people don't actually exactly know the answer. Okay, so where some of these other things, such as lung cancer, colon cancer, breast cancer, we have clear, we see the association, and people have done experiments, and they can basically clearly delineate that as a basis. For stomach cancer, there are hypotheses, there are ideas about why that happens, but no clear definitive reasons. But well, I'll give you a couple of those, couple of the things that people think underlie this. So one of the things that people believe is that one thing you can see here is that in green, basically you can see this colon and rectum rates. So this rates of colon cancer were actually much lower down here. So they basically have gone like this. So while we have had an increase in rates of distal gastrointestinal cancers, such as colon and rectum, we've had a decrease in rates of upper gastrointestinal cancers, such as the stomach. And one of the reasons people believe that the reason for that is that they believe that it's due to two things. And well, one of the main ones is that they think that the increase in colon and rectum cancer rates is due to the Western diet that we have. So basically, an increase in foods, dietary reasons, right? So fatty foods, burgers, fried foods, etc., decrease in vegetables. And one idea is that the Western diet can increase the rates of distal cancers, and it can, in the US, has actually led to a decrease in the rates of stomach cancers. That's one other possibility. The other possibilities are incident rates of H. pylori infection, Helicobacter pylori infection, and how that can interact with the stomach. We really don't exactly know what's the underlying reason, but there are hypotheses. So that's a really good observation. Here's a chocolate for you. Next question.
Is like leukemia and like lymph node cancers more likely to spread than solid cancer cells?
Okay, that's a really great question. Oh, what's your name and where are you from?
I'm Cyrus. I'm from Great Neck South.
Okay, great. No word up to his high school, guys? What? All right, good. No. Okay,
They don't go to my school.
so the question is, are leukemias and lymphomas more likely to spread than solid cancers? Okay, so leukemias, by definition, they're in the circulatory system, right? They're floating in our bodies. We have them in our bone marrow. We have them in our blood. Someone with leukemia, by definition, these cells are within the circulatory system, and they can spread throughout the body. They can be present throughout the body. They don't. So in terms of their spread throughout the body, yes, by definition, because they're in our bloodstreams, they're throughout our body and they spread. We don't call that metastasis because, unlike solid cancers, they typically don't leave the bloodstream and grow out in certain organs. Okay? What they typically do, the reason they cause death is by occupying space in the bone marrow, getting rid of these other blood types. But yes, so that is actually the case. So you're going to get one of these. Next question.
I'm Keisha from Aquinas High School. And I know you had said that HPV causes cervical cancer in females, but hasn't been seen to cause any cancers in males?
Yeah, so that's a really good question. So, excellent question. Anybody know the answer to that? Well, let's take a vote. Do you guys think HPV can cause cancer in males? Yes, it can. And so it can actually cause, it's been basically implicated in both penile cancers in men, but also anal cancer. So yes. So great. So here are your chocolates.
Oh, wait, I have another question. Oh.
Next question. Oh, sorry. Yes.
Yes, my name is Terry from William Cullen Bryant High School.
Okay.
And
Yes,
my question,
yes.
my question is, if we have the information and documented that PS53 and the
Retinoblastoma.
yes
And P53.
are tumor suppressors, why can't we replicate that and sort of create a cure for cancer?
Okay, I think that's a really great question. So she's saying let's take it to the next level. We have this information, and why don't we try to use them therapeutically? And actually, there are many scientists that are trying to do that. And what they're doing is they've tried to put these back into cancers. That's the idea, or turn them back on if they've been turned off. And so people are actually working on that. That kind of approach has been a little bit difficult because it's kind of in the realm of gene therapy, which is to try to replace a gene that is defective by using things such as a virus. And so it's been difficult and it's been challenging. But there are investigators out there who are trying to deliver P53 back into cancer cells. And if you have in certain experiments that were done, when individuals had a cancer that's formed in a mouse, and they had a way to turn it back on in that mouse, they found that the tumors can form, and when they turn it back on, the tumors can regress and totally be shut off. So in the mouse model of cancer, people have done that type of experiment where they've turned the gene that's been shut off back on, and it can cause the cancer to shrink back. It's challenging to go from that to human patients and therapeutics, because you have to somehow find ways to deliver those genes without using viruses that can themselves cause cancer. But I think that's a very, very good and that's a very good question. Pushing the envelope. I think this is the last. All right, that will be the last question. Yes.
Hi, I'm Marina from Southside High School. What is it about stem cells that are can really help in I guess curing cancer or in the
So is there something about stem cells that can help us?
What is it about them? I guess.
So actually, what I would say that the best way that stem cells can help us get towards a cure in cancer is to allow us to study them, because there are certain features that that are shared between stem cells and cancer cells, such as their undifferentiated states. So stem cells can give rise to a number of different cell types, okay? And so they're less specialized, they're less differentiated, and that feature is a feature that's in common with cancer, okay? So biologists, by understanding what allows them to be undifferentiated, or understanding what are the growth factors that these stem cells need, have found that many of the ways that stem cells in the normal body operate those features are common to cancer. And by understanding that, we may be able to block some of those activities in cancer and get therapeutic efficacy. So great question. Last question, and then it's pizza time. Okay. Yes.
Hi, my name is Ariella. I go to Meyer. I was just wondering why some cancers are more deadly than others.
Okay, excellent question. So she asked, why are some cancers more deadly than others? It's a very good question. So an example of this is, for example, testicular cancer, okay, what Lance Armstrong had. It's actually extremely, extremely curable. So individuals can have testicular cancer, and that cancer can actually spread to different organs. And actually, those patients can get chemotherapy, and they can actually be cured of their cancer. And those cells are very sensitive to these chemotherapeutic agents. However, pancreatic cancer is extremely aggressive. And even when caught at early stages, it can spread rapidly, and it can kill the individual. To be honest, we really don't know the basis for these differences in the ability of certain cancer cells to kill patients. One of the things we know is that there are certain cancer types that are extremely sensitive to chemotherapeutic agents. Some of those cancer types are actually cancers that grow very fast. So an example would be, for example, testicular cancer or certain lymphomas that can grow very, very rapidly. So on the surface, it seems that they're going to kill the patient in a few days because they're growing so rapidly. But by that feature that they grow so rapidly, they're also very sensitive to chemotherapy. Because they're running so fast, if you hit them with chemotherapy, they suddenly fall apart. But in terms of other cancers like pancreatic cancer, why is it so aggressive? Lung cancer, why is it so aggressive? Relative to, compared to something like cervical cancer, which even when caught at sort of later stages, it's curable, we don't know. We don't know. Some people say that it could be due to the spectrum of mutations that they inherit. pancreatic cancer, melanoma, et cetera, might inherit many, many mutations, but things such as cervical cancer might be caused by a viral infection inactivating a certain small subset of genes. That might be the case. All right, last question. She's been.
Thank you.
And you guys are all gone.
My name is Sana Siddiqui. I'm from Tenafly High School.
Excellent.
Right, Okay.
Yes?
So there are a lot of warnings about foods being possible carcinogens, as in possibly causing cancer. So I was wondering how they come up with those warnings and how they figure out what kind of foods could cause cancer.
Okay, I think that's a really great question. It's a little complex. I think it'll take more time to answer, but I'll give you a simple explanation. So one of the a really great scientist called Bruce Ames at actually University of California, Berkeley. Come on, guys. Yeah. So he actually developed an assay to determine whether certain compounds are mutagenic. Like I told you, cancer results from mutations in genes. So the notion he had is, well, how do we figure out what foods cause cancer? He said, maybe I can devise a system that can tell me what foods can cause mutations, and then I can infer whether those compounds can actually cause cancer. So he created a cool test called the Ames test that involves using bacteria, and where you can basically grow them on a plate, and if they experience a mutation in a gene that you can visualize, you can see, you can basically visually see the mutation rate of a chemical compound. And he used that assay to generate a list of how mutagenic certain compounds are. And so that is one way in which we can know whether certain compounds are mutagenic. Now, that doesn't necessarily mean they're carcinogenic, okay? That sort of suggests they are. That was a cool assay because he actually showed that many things that people thought were mutagenic were not mutagenic, and he showed that other things that people had no idea are mutagenic are highly mutagenic. Another thing that we can do is, as I told you, epidemiology can be very powerful. So you can actually look at large populations of humans, and you can actually ask the question, what are these people doing? What are they eating? And what is the rate of cancer formation? And one of these studies is called the Nurses' Health Study, and this is done in Boston at Brigham and Women's Hospital. And what they basically do is they ask, they take thousands and thousands of nurses, and they have them fill out forms throughout the year, describing all the things they ate and all the physical activity that they participated, whether they smoked or not, and they follow them for many, many, many years, And then they basically ask, what is their incidence of cancer? And they can come up with sort of strong statistical correlations between them eating certain things or not having certain vitamins or having certain lifestyles and the incidence of cancer. And then those compounds can then be tested in mice, okay, to see if they indeed are causal. All right? And then those lifestyle changes you can do a trial to eliminate those compounds from lots of people, and you have a control group and an experimental group, and you can see if performing that intervention actually reduces cancer rates. So it's involved, but those are sort of how we arrive at these, people arrive at these conclusions. It can take a lot of time, but it's very important to do. And so that's why it's sort of important to think about things that one puts in their mouth or things that you drink. Every year, there are lots and lots of new things that come up, and drugs and compounds, and it could take us a long time to figure out that those things are actually bad.
Thank you.
All right, great job. Pizza time. So we talked about cancer the first half, okay? And I hope we impressed upon you how fun it is to be a cancer biologist, and the importance of understanding a process before trying to treat it, okay, to understand the nuts and bolts. In the second half, we're going to move from something that's complex to something that's even more complex. So we talked about cancer the first half. In the second half, we're going to talk about metastasis, or the spread of cancer. So I'm going to start this off with another animation to wake you guys up. Actually, I lied. We're going to start this off with a little overview. So in the second half, I'm going to talk to you a bit about the bigger picture, the overview on metastasis. We're going to talk about historical work. It's always important to know history. You know, I know you guys are science students. I always liked science more than I liked history, but actually, it's really important to know the history of things before trying to advance certain areas. So it's important to know about the people that came before you and what did they learn, and were they successful? What are the mistakes that what were the mistakes that they made? How did they conquer the different areas? We're going to talk about cellular studies involving metastasis, and we're going to give molecular examples of how metastasis occurs. And so I'm very interested in this area because that's the area that I study, so cancer metastasis. And after that, we're going to talk about cancer prevention. So we're going to really train you guys to be junior oncologists and junior physicians. And then we could, if we have some time, we'll also have some question and answers, a little question and answer session. Okay. So remember, I showed you this slide, which showed you the death rates from cancer in 1950 and 2003. And one thing that I wanted to impress upon you is that, as you see here, there's this really high rate of cancer deaths. And the question that I came up with when I was trying to figure out whether I wanted to be a neurobiologist or a cancer biologist was, I started thinking about this and wondering, what is it about cancer that kills? And as I told you guys, the majority, the main cancer type that humans get are epithelial cancers, these solid cancers. And we also know that the way that these solid cancers kill individuals is by spreading from one organ to the other. So the vast majority of deaths due to cancer result from metastasis, or the spread of cancer. Many of you have heard people who have died of cancer, you've heard terms such as the cancer came back or the cancer spread. When they say the cancer came back, in those patients, it never really left. It didn't disappear and then reappear, right? The cancer was in that individual, the cells spread to another organ, you couldn't see them for a few years maybe, and then they grew up in an organ, and then you could see them, and then they did damage. And you can see that metastasis really is the cause of death here. So this is the survival probability for women diagnosed with breast cancer. At time 0, 100% of them are alive, and this is a function of years. Women who are found to have some spread of their cancer, such as spread to a lung or liver or brain, you can see that their survival probability plummets, so that by about 9 years, less than 17% of them are alive. Okay? Women whose breast cancers are diagnosed but are local, so they don't have any spread to lymph nodes, they're just confined as a cancer that's just localized to a certain area, they do very well. You can see that by 10 years, more than 90% of them are alive. And then women who have spread to their lymph nodes are somewhere in between, because the cells that are able to go to lymph nodes also have capacities that allow them to spread even further. So this is a little cartoon about sort of depicting metastasis. It's a little bit weird because it looks a bit cartoonish. So this is the primary cancer, and it's basically showing that within the primary cancer you have cells, And these are cancer cells that have been, and you can see that these cancer cells are migratory. If you look at them under a microscope, you can see that they move. They're not happy just sitting around. And you can see they don't really care about whether they're bound by their neighbors or by other cell types. They're cruising around. And this is what happens. From the primary tumor, cells enter the bloodstream. That's called intravasation. Into, vasation is the vessel. And these cancer cells float in the bloodstream, okay? And as they get in the bloodstream, they face all sorts of different threats. You have all these different cell types. In white, you have immune cells. In red, you have red blood cells. The hemodynamic forces push these cells against vessels. The cancer cells can undergo death. They can die from these shear forces. And some of these cells get recognized by immune cells, for example. And immune cells of the immune system can bind them, and they can kill them, okay? Some of these cells can actually eat up the cancer cells, and they detect them because they see that they're abnormal. They don't look like normal cells. And other cells get away. They leave the blood vessel, and they come out of the blood vessel. They go into the organ, and that can be the liver, the brain, the lung. And once they go there, they expand. They start dividing uncontrollably. And that's the metastasis. And that's what we're going to talk about during the second half. So to show you an image of metastasis, what we have here is a PET scan. Now, this is not a scan of your pet. It's actually a PET scan. Thank you. I really thought about that over the weekend. So what this is it allows you to visualize cancer cells throughout the body non-invasively. That means a person can be given something in their bloodstream, and it's actually a form of glucose that can emit positrons, and the person can be placed into a detector, just like you guys have seen people going into MRI machines or CAT scans. And then cancer cells take up this glucose very actively, okay? And because they do that, you can see them, and you can visualize them. And you can see that this individual has signals throughout their bodies, so by their bones and their spinal cord, some signal below the liver. And these are metastases where the cancer has spread. Okay? So metastasis was coined by Jean-Claude Recamier in 1829. It's defined as the transfer of cancer from one organ to another, not directly connected to it. So this is Jean-Claude, and he was a martial artist. Actually, I'm joking. This is Jean-Claude Van Damme. I actually couldn't find a picture of Jean-Claude Recamier, so I decided to put another Jean-Claude here. But now this Jean-Claude Recamier could have looked the same and could have been equally as athletic. I really don't know that, but Jean-Claude Van Damme. Okay, so metastatic progression is a complex multi-step process, okay? So it's pretty amazing, actually. So you guys just have to think about all the things that a cancer cell has to do to get from this point to this point, okay? Now, first, as I told you, as we learned, cancer cells must acquire mutations in oncogenes. So basically, they must activate oncogenes. They must silence tumor suppressors. That gives certain cancer cells these aggressive phenotypes. Aggressive phenotypes include the ability to migrate, to move, to chew up tissues, to migrate through tissues, to basically grow blood vessels here. These aggressive cells can actually detach from each other, and they can enter the bloodstream, okay, and go on a trip. Just like many of you are going to go on a trip to college, these cancer cells leave the home, the comforts of the home, and they get into the bloodstream. Once they get into the bloodstream, they have to survive these forces, hemodynamic forces. They have to survive being somewhere that they're not used to without all the comforts of the organ that they spent their lives in. And they flow through the bloodstream. And many, many, many cancer cells are killed in the bloodstream. Okay? Some of these cells are actually able to attach to blood vessels near organs. And some of the cells, even fewer fraction of those, are able to move out of the blood vessels. Okay? And a rare cell can actually move out, many of these rare cells can move out of blood vessels and get into organs. Now, these cells are in an organ that looks completely different than what they're used to. All their programming tells them that this is not where they should be. They're not used to the environment. And it could take them days, months, years to sort of adapt and have what it takes to grow in that organ. And the colonization of that organ, or the expansion of this single cancer cells in this organ, such as the liver or the brain, that's called really metastasis. Okay? And cells must be able to move through this tissue. They must chew it up. They must degrade it. And that's what causes the death of the patient. It's called organ failure, right? The liver no longer works. The brain no longer works because the tissue has been destroyed. So concepts that I want you to think about during this talk are the importance of diversity and heterogeneity. I think as we look around, as you guys look around the room, I don't have to convince you about the importance of diversity. You guys are all diverse, different backgrounds, different beliefs, different countries of origin. And obviously, this is really great, and we enjoy it. And it's very important because it creates for a productive society that has many different attributes that one individual society with a homogeneous population wouldn't. Okay, and that's one of the reasons why America is so great. That's also one of the reasons that cancers can metastasize, because individual cells within these cancers, as they expand, they can be diverse and they can be heterogeneous. Some of them can do one thing, others can do another, and a very rare fraction can do many things. Okay? The concept of sequentiality, okay, sequential. Metastasis is a sequential process. As I told you, the cancer cells must leave the primary tumor, get into the bloodstream, survive in the bloodstream, attach to the vessels, get out of the bloodstream, and they must grow in that organ. So this is sequential, okay? I'm going to talk to you guys about selection. So you guys are familiar with selection, such as the notion of natural selection that was put forth by Charles Darwin. But here, selection, it works in the same way, where the selective barriers are each of the stages of metastasis. Okay? Each stage is a selection. Just like for you guys, going from junior high to high school was a selection. Getting into an honors class or AP class is a selection. Getting a 5 on your AP exam is a selection. Getting your 1600 on your SAT is a selection. Getting into college is a selection. Getting into a Ph.D. program is a selection. All those are selections, right? And they just narrow down the field. And metastasis is the same way. Every stage is a selection. And the cells that are left over at the end have already done a lot of things and displayed a lot of aggressive, malignant features that can actually help them in the last stages, right? Just like your calculus that you learned in high school can help you in your physics class in college, same idea. The last notion is dormancy. So cells don't have to do each of these sequential things immediately. Sometimes cells can just sit around in an organ for many, many years. So that's kind of one thing that's scary about cancer, is that certain individuals can have cancer and they can have the cancer taken out, but many years later the cancer can metastasize. And that's because a lot of these cells can actually lie dormant inside organs for many years, undetectable, and then they can basically expand at a much later time. And so we're going to talk about some of the historical questions that have come up. So if you guys think about it, how did people historically view metastasis? If you think about it, we didn't know anything about science or cancer, and we had individuals in society that developed cancer. You might see someone having a tumor on their breast, and then that tumor grows, and then many years, let's say 3 or 4 years later, you suddenly see that person has something in their brain, has something a mass growing in their liver. It's not, back then historically, it wasn't necessarily immediately clear how this happened, right? That individual cells from that primary site went to those other sites. Someone might think that all these other tumors expanded independently, and they weren't related to each other. But so let's think about some of the historical questions. One of the questions that people raised was whether cancer cells in a primary tumor, whether every cell in a primary tumor has the same capacity to metastasize. So we have a tumor that expands. There are many, many, many cells in that tumor. There can be millions of cells in that tumor. Do they all have the same ability to metastasize, or do they have distinct abilities to metastasize? The other question is, do all the metastases in a given patient, let's say someone who has a liver metastasis, a brain metastasis, a bone metastasis, do they derive from different parental cells, different or original parental cells, or do they come from a common cell? The next question was, do cancer cells spread randomly, or is there some bias to the organs that they spread to? And finally, and potentially most difficult question is, what are the molecular drivers of cancer metastasis? Just like we talked about in cancer formation, these cells do things, are involved in processes and have phenotypes, basically, essentially because molecules inside them, these proteins, are mediating these phenotypes. So first question, do all cancer cells in primary neoplasm have the same capacity to metastasize? So one of the leaders in the field was Josh Fidler, who is a veterinarian and also a researcher who's really contributed a lot to our understanding of metastasis. And what he did was he addressed this question, whether individual cells in a primary tumor have the same capacity or different capacities to metastasize. And the answer he came up with was that no, neoplasms, these primary cancers are heterogeneous. How did he do this experiment? What he did was he took a mouse which had a cancer, so melanoma, and he took the melanoma out. He took the cells from that melanoma, split up the cells, and took one cell, put it in one plate, one cell, another plate, and he split up these cells into individual cells. He expanded these cells, made individual clonal expansions of each of these cells, and then he injected them into subsequent mice. Okay? So what are the two possibilities that one would arrive at? If every cell had the same capacity to metastasize, when he injected these cells into these mice, they should all get metastasis, or they should all not get metastasis. What he found was that once he injected these cells into these mice and looked 2 months later, some injections failed to form any metastases. The mice lived and there were no metastases formed. Other cells, when injected, formed a modest amount of metastases, so these nodules that you see here, small nodules. Other cells were very dramatic in forming metastases, and you can see it, sorry, you might be able to see it down here, okay? So that showed that cancer cells within the primary tumor are heterogeneous. They have different capacities to metastasize, okay? And so this is what it looks like. There are different cells within the primary tumor with different abilities to metastasize. And the other cool thing he found was that he actually quantified the ability of these cells and asked, out of all the cells in the tumor, that when you inject them, when you inject all these cells, let's say you take the parent population, what percentage of those cells leads to a metastasis? And he came up with a very small number, 0.01%. Okay? So 0.01% of the cells could form metastases. That tells you that this is a very challenging process. Okay? It's a marathon or an Ironman. So Fiedler also did other experiments. Fiedler and others have done additional experiments with human cells. So what you can do is you can take cells out of human patients. So here you can take cells from a woman with breast cancer, and you can do the same experiment. You can take her cells, make single cell isolates, expand those, and inject them into mice, and you see the same thing. Okay? The same thing happens. Cells within humans are also heterogeneous in their ability to metastasize. So the next question is, how do we generate that heterogeneity? How do we help metastasis along? And we generate that diversity by having more cells. We create that diversity by having more cells and larger tumors. And that is shown here very simply in that tumors that are larger are more metastatic. Tumors that are larger, these are tumors that are greater than 10 centimeters in diameter, are more metastatic than tumors that are smaller. And the reason for this is that these tumors have more cells. Okay? They have more cells that can participate in the process. Okay? And they also have more diversity and heterogeneity so that they have a higher likelihood of having the cells that can complete the task. So this is our conclusion. Cancer cells are phenotypically heterogeneous. The next question that was historically addressed was whether all cells within a given metastasis derive from a common progenitor cell. So let's say you take a single metastasis in a patient. Did that arise from a single parent, or was it that multiple cells landed there and they worked together and they formed the metastasis? So both things are possible. And I'll give you examples of historical experiments which suggest that in one system anyways, that it actually looks to be a single common progenitor. And these were experiments done by Talmadge and Fiedler in 1982. What they did was to have mice that develop a melanoma. Okay, so this is a skin cancer. They irradiate that tumor and form, when you do that, you form genetic changes in each of the cells that results in alterations in the chromosome numbers. And so each cell can be marked by a different genetic lesion. And so one can then take those cells and look at them under microscope and look at the karyotype and see differences and be able to have an identity of two different cells. And then they took these cells, which are now marked differently, injected them into a mouse, and then at some point afterwards, a metastasis formed. Then they take that metastasis out, and they take all those cells and they ask, do they have the same genetic lesion? Do they look similar in terms of their chromosomes? Or do they look like their siblings? Or they're really not from a common parent, they're different. And what they found was that those cells actually had the same chromosomal alterations, consistent with that metastasis having arisen from a common parental cell. Okay, so these types of studies have been extended to humans. So in these experiments were done by Liu and Bova recently and published in Nature Medicine. It's a little complicated, so I'll try to simplify it. What they did was they took men with prostate cancer. Okay, so this is a bone scan of a man with prostate cancer, and these black dots are metastases. Okay. So the prostate is here, and they're basically making a point at sites where they picked off metastases. So in this gentleman, they have 4 different metastases that they've taken out. And they've looked at the genome of this person and asked, what do the chromosomes look like? And by doing that, they can ask, are they similar to each other or are they different? Did they come from a common parent, or did each of the metastases arise from different cells within the primary tumor? And what they found was that, I guess you might be able to see here, is that this metastasis had a region of chromosome 5 that was lost. So did this other metastasis. So each of these metastases had the same region of chromosome 5 that was lost. Okay? And they found a similar thing with chromosome 8. All of them had the similar regions of chromosome 8 that were lost and common regions that were gained. Okay? Now, that's not the entire story, but so what does that tell you? That suggests that there seems to be evidence for a common progenitor. But there are also some subtle differences between these metastases, which could have arisen after the cells have basically arrived in those end organ sites. So you can see they did this with a number of men, but basically all of these individuals had prostate cancer, and they found similar things in all these individuals. So there are two possibilities for that, how that can happen. One possibility is in the primary tumor, you have heterogeneity. You got these cells, these brown cells, blue cells, green cells, and the cells spread, okay? And certain cells that have spread to certain organs can actually survive in those organs and grow up. So one possibility is that these blue cells land in the liver and they expand. And then some of the cells some of these blue cells in the liver then enter the bloodstream again. And then they spread and form new metastases in the brain and the bone. And then they all look the same. They all look like they have the same genetic backgrounds. So that's one way. That's metastasis from a metastasis. The other possibility is that in the primary tumor, this cell got into the bloodstream out of the primary tumor and was very, very effective at forming metastases. And this one spread to a bunch of different sites, and all of these daughters survived, but none of these other guys survived. Okay, so those are two different possibilities. But the general conclusion is, we have two conclusions. One is that the cells within a metastasis seem to have arisen from the same common progenitor cancer cell. And this is in melanoma, based on the experiments done in melanoma, and that macroscopic metastases, so these large metastases, appear to arise from a common cancer cell progenitor. So the next question, this is one of the this is an interesting question that's been a big issue in the field for over 100 years, and that is, do cancer cells metastasize in a random fashion? So do they go to organs randomly? Do they spread to organs randomly? Or are there some preferences with respect to which organs they go to? Do cells decide, do they have a preference for certain organs as opposed to others? So in 1889, Stephen Paget basically proposed the seed and soil hypothesis, okay? And he made observations in cancer patients. And for example, in breast cancer patients, he found that the likelihood of them developing metastases to certain organs was not random. So women seem to get bone metastases and lung metastases at higher rates than brain metastases. They formed, 70% of the women had bone mets or lung mets of the women with breast cancer, and they almost never saw metastases to the spleen. Okay, this is a spleen. Smiley face for you guys. Okay, so that was the seed and soil hypothesis. The point was that organ dissemination is non-random and depends on the compatibility between the seed, the cancer cell is the seed, and the soil, and that's a target organ. So I guess he had a quote that said something along the lines of, the wind can blow the seed in all directions, but the seeds will mature and flower when they fall upon congenial soil, okay? So that means that something about that organ, those organs, is favorable for these cells to grow. In 1929, James Ewing came up with another model. He said, you know, I don't think Paget's got it right. He put forth the vascular supply model. This is James Ewing, no relationship to Patrick Ewing of the Knicks. Does anyone know Patrick? He was a dominant player. Yeah, I saw a hand raised there. Excellent, excellent. So he proposed that, look, it's random. The reason you see non-randomness with respect to which organs develop metastases is simply the blood vessel supply, okay? So you see more metastasis to certain organs. For example, colon cancer goes to the liver because it's connected to the liver via the circulation, the portal circulation, okay? And so this is what he's proposing. Colon cancer goes to the liver. That's the primary site that colon cancer goes to. And secondary site is the lung. The circulation for the liver goes up north to the lung. So you go from here, you go up north to the liver, north to the lung. Makes sense. But sometimes you can find data that supports your hypothesis, and this did in this fact, but you should also look at the data that doesn't support your hypothesis and see if it fits. And the data that didn't support his hypothesis was that the kidneys get a huge amount of the blood supply, right? But we rarely get metastases to the kidney. For example, in breast cancer patients would rarely get kidney metastases. The spleen also gets a lot of blood flow, but you rarely get metastases to the spleen. So Ewing's model didn't exactly make sense. So how was this reconciled? Well, it was reconciled in two ways. One, Weiss and Sugarbaker in 1979 did an extensive review of human autopsies, over 700 autopsy cases, okay? They looked at all these individuals and asked, who had died from cancer, and basically asked, does the data support Ewing's model or the Fiedler model? Is it random or is it non-random? And what their conclusion was, there was clearly a display of organ bias. Certain organs, such as the bone and the lung, were much more favored, and certain organs, such as the spleen and the kidney, rarely got metastases, despite similarities in blood flow. So one of the other interesting things about science is sometimes data comes out of experiments of nature, so experiments that so there are information that can be gained from things that are just done for other purposes, that weren't done for the purpose that you were interested in. So this is what happened with this individual, Taryn, in 1984. And how this experiment happens is that there are certain women who have, for example, women with ovarian cancer. You guys may have seen this. They develop what are called ascites, and sometimes people with colon cancer can get them. But for example, women with ovarian cancer can get bloating of their bellies, and the ovarian cancer can grow inside the peritoneal cavity and lead to fluid accumulation. This fluid can be really cause a lot of discomfort for these women, and expand their abdomens. And so what Taryn did was to say. Actually, you can go and stick a needle in and drain it, and you can keep doing this, but the patients can actually get infections, and it can be painful and whatnot. And so what Taryn did was he said, why don't these women are end-stage women, and I want to make them more comfortable. So what he did was he created a shunt. And what that is that he placed a tube from the belly, right, peritoneal cavity. He took that tube and he connected it to the jugular vein. Okay. Sometimes doctors and surgeons do really outrageous things just because they can, and sometimes people will say, "I can't believe this person did this," but sometimes everything goes okay. And in this case, it did go okay because a priori thinking, why is this person making a tube from the belly, taking ovarian cancer cells that are confined to the belly and connecting it to the vein? where they could suddenly go into the heart and spread out throughout the body, right? It sounds kind of crazy, but that's what he did. And what this does is this decompresses the belly. So a lot of these women feel more comfortable because it gets rid of the fluid, it decompresses the belly, and that's just inside them. But what did he find? He found that when he did that, one possibility is that you're injecting millions and millions of cells into the heart, and that's going into the lung. And, that doesn't sound like a good idea. But when he did that, they found that none of the patients ever developed lung metastases. The women all ended up dying from the cancer within their bellies. Even though you're injecting millions of ovarian cancer cells into the circulation, these women didn't get metastases to the lung. They didn't get metastases to the brain. These ovarian cancer cells didn't care so much for the lung or the brain. So this was sort of an experiment that was not done to support Fiedler's hypothesis, but it clearly does support his hypothesis. So, so further experiments were done by Hart and Fiedler, and what they did was they took melanoma cancer, and what they did was they injected the melanoma cells into mice, okay? And they found that if they look 3 weeks or 4 weeks after these mice have been injected with melanoma, and they ask, where are these cells? They find that the cells go everywhere. They're present in all sorts of capillaries everywhere. They can be found in the kidney, they can be found in the liver, they can be found in the spleen, they can be found in the lungs, brain, everywhere. But if they let it go, so initially they have the melanoma, they put it into another mouse, the cells go everywhere initially, but if they allow 2-3 months to go by, only the cells in certain sites, such as the lung or the brain, grow out in those sites. Okay? So, and all these other cells at these other places, they basically die off and are unable to grow up. Okay? So this was experimental support of organ selectivity in melanoma and in ovarian cancer. Okay? Now, the same experiments can be done with human cancer cells, and these are experiments done experiments done by Fiedler, but also Andy Min and Yibin Kang in the Massey laboratory at Memorial. And so what was done here was that you can find a patient who has breast cancer with metastases to bone and lung. You can actually take her cells out of the fluid lining her lung, okay? And you can actually take those cells, which we've already talked about are heterogeneous, inject them into an immunodeficient mice. And what's pretty amazing is that these mice, if allowed enough time, will also develop lung mets and bone mets, just like this woman had, lung mets and bone mets, okay? So this shows you that this organ selective feature of these cells is retained in a whole different organism, okay? And this is, again, experimental support for organ selective tropism. Another experiment that was Josh Fiedler did, which was cool, was this question of, well, okay, the cells go to the lung or the bone. Did that ability exist in the primary tumor? Or is it something that they adapted to after some time? Okay? And what he found was that that capacity to metastasize to the lung or the bone was present in those cells in the primary tumor. And how did he do those experiments? He took cells from mice, but this can also be done was done in human cells. Those cells are taken out. Again, single cell clones were made. You take single cells, you make single cell isolates, and then you expand those in a dish. You inject each of those into the mice, and what you find is that one single clone might not metastasize. Some single cell clones, like the bone, they metastasize to the bone well. Others metastasize to the lung well. Okay? So this is work done by Fiedler, but also by Andy Min and Yibin Kang in the Massey lab. So cells, certain cells, okay, we've talked about melanoma and breast cancer, and ovarian cancer have preference for certain organ sites, okay? And that preference appears to exist in the primary cancer initially. Okay, so in conclusion, we've talked about how metastatic patterns are not random. We've talked about the fact that vascular supply, look, Ewing was not entirely wrong, okay? And that's another thing with respect to science is that, you know, people can put forth hypotheses. Some hypotheses might be better than others, but sometimes the ultimate answer ends up being a sort of amalgamation of multiple hypotheses over time. Historically, one might win over another, or sometimes the right person doesn't win at that time, and then 100 years later, people rediscover that individual if they're lucky. But a lot of times, it's a mixture of multiple ideas and hypotheses. Ewing's idea of this being random and it's all vascular supply was not correct. However, vascular supply can shift the probability distribution simply because based on the vascular supply, the cells that leave the primary tumor have a higher chance of hitting that organ because that's the first organ they hit and they land in that organ than other organs. So for example, for gastrointestinal cancers, that's what seems to be the case. The origin of where a cancer cell arises can impact the tissue to which it'll metastasize to. So this is something someone brought up earlier about different cancer types. So certain cancers, such as breast cancer, for many women with breast cancer, this cancer likes to spread to the bone or the lung. For melanoma, you get lung and brain metastasis, for example. For other cancers, such as uveal melanoma, you don't get bone, you primarily get liver. For men with prostate cancer, when the cancer spreads, you typically get bone, and sometimes you get some lymph node spread. So you don't get brain as much. So depending on where the cancer arises, there are different propensities for metastatic spread. And finally, that organ metastatic biases pre-exist in the neoplasm and can dictate metastatic outcomes. So what I want to do is now just talk to you guys about how these cells metastasize, okay? Now, this is really an interesting problem, but it's fascinating, but it's, as you can imagine, scientifically difficult to study because if you think about many other aspects of biology, many aspects of biology are very focused, and you're studying a single process, and you can find the molecules that mediate that process, and they're more tractable. Here, metastasis, as I told you, is very complex. It's multi-step. It takes time. It requires an animal model, and it requires, you can study things in one cell type, and they may not necessarily relate to the genes involved in another cell type. So this field is sort of relatively new in the molecular understanding of metastasis. So one of the questions that has come up with respect to the molecules involved in metastasis is whether metastasis is caused by single genes or mediated by the cooperation of multiple genes, okay? So is there a master gene that in every metastatic cell turns on and allows that cell to metastasize, okay? And what we know from both human studies and experimental studies is that metastasis requires the concerted activity of many proteins and many genes, okay? It's sort of like similar to a corporation. In order for it to get to a final product, many expertise need to be put together to achieve this complicated task. And so the human data, some of the human data comes from the fact that over the last 15 years, technologically, we've been able to develop tools that lets us survey all of the genes that are known to be expressed in human cells. So some of you may know about this. These are called gene chips. And what these gene chips do is they let you look at all the messenger RNAs that are present in a given cell type, okay? So then what you can do is you can take an individual with breast cancer, you can take a woman who has breast cancer that doesn't spread, and you can take their tumor out, and you can extract the messenger RNA, and you can put it on the gene chip and see all the genes that are up and all the genes that are not that are down. And then you can take another woman's breast cancer, in this case, a woman whose tumor has metastasized, and you can ask the same question. And when people have done that, they found what they found was that women whose cancer spread, those tumors have turned on many genes that the tumors of the women whose cancers don't spread have not turned on. Okay, so it looks, and you can take tumors without knowing whether that woman is going to spread or not, and you can take their tumor and ask what happens to these 40 genes. And you can say, oh, this woman's tumor has high levels of these 40 genes. This woman does not. And you can predict, okay, the likelihood that that woman's cancer will spread or not. This is not perfect, of course, but it tells you that groups of molecules are mediating this process. Okay? So how do we identify the genes that mediate metastasis? So these experiments were really originally, these experiments were pioneered again by Josh Fidler. And he was doing this stuff in 1973, a long time ago. And these are really cool studies where what he did was he said, look, I can take cells from a mouse or a human, and I can inject them into an immunodeficient mouse, and they metastasize. Okay? What if I took those metastases and took them out and re-injected into another mouse? What if I kept doing that? Could I get super metastasis, cells that are super metastasis? So I told you these cells are heterogeneous. Okay? What if you inject them into a mouse? Okay? And you get metastases. What if you take these cells out? Okay? And what if you keep re-injecting into other mice? Can you get super metastatic cells? And that's what happened. Initially, you get very small nodules. You take those out. You inject it into another mouse. And this is the lung of the mouse. Okay? You can see you start getting larger nodules. And then you inject that into another mouse, and you can see that the entire lung is now occupied by metastases. And you can see this in the mice because you can put a reporter gene in the mice, and you can visualize them just kind of like the same way you do a CAT scan. And you can see this last mouse has really bright signal, showing that the cells these blue cells that you've extracted, you've really enriched for them, that you've actually been able to purify from this original parental population the super metastasizers, okay? So then you're like, okay, so what? Andy Min and Yibin Kang and Jean Massey's lab, what they said was, well, let's take these and ask what are the genes that are up in these cells, okay? So now you can do that. You can use these gene chips again and ask, what are the super metastasizers doing that their parents can't do that well? What are the super metastasizers doing relative to the poor metastasizers? What genes are up in them relative to these guys? If you can find that, maybe you can then take away those genes from the super metastasizers and keep them from metastasizing. Everyone's on board? Excellent. And so when you do that, you can take cells that go to the bone. You can see the mouse, its bones have metastases here. It's the skull here. It's the femurs. Here, this mouse has only lung metastases. And you can see they have a fracture in their bone here. And this is their lung. It's totally occupied by metastases. And when you look at the gene chip, you see that the cells that go to the bone have bones that are used by cells in the bone marrow, for example, or by cells in the immune system that allow normal cells to get into the bone marrow, okay? They also activate genes that let cells break down the bone, okay? So these are genes that activate osteoclasts. You guys might have learned about osteoclasts. They're the cells that chew up bone, and when they do that, when your calcium levels are low, your body activates osteoclasts. They can chew up bone and release calcium and other good stuff. What the cancer cells do is they trick those cells into chewing up the bone and providing growth factors for them in that, and opening up space for them in the bone so they can break down the bone. And then lung metastatic cells activate genes involved in restructuring the blood vessels of lung. Okay? So the fourth thing that we know from a molecular level is that multiple gene products are required for metastasis is now, if you take all those genes that we found on the gene chip, and you take a cell that can't metastasize, and you put one of those genes in, not much happens. But if you put 3 of those genes in together, the cells become much more metastatic. Okay? So these are these experiments. This is the metastatic activity on a log scale. This is weeks after putting the cells into the mouse. The blue is the poorly metastatic cells. You put a single gene and you get a little bit of an effect, a little bit of effect. But if you put 3 genes in, you get this big jump in their ability to metastasize. So multiple proteins participate in metastasis. So this is an overview of what happens in metastasis. So at every step, many genes are required for cancer progression. So in the tumor itself, you have oncogenes that are active that we talked about. Remember, guys? Things like the epidermal growth factor receptor, PI3 kinase. You also have a loss of tumor suppressors, such as P53, P10, RB. That allows a tumor to form. And then you have genes that allow cells to detach from each other, to enter the blood vessels, survive in the blood vessels, get out of the blood vessels, and then genes that allow cells to ultimately break down those organs. And again, remember that there's this concept of dormancy. Cells can sit in a tissue, such as an organ, for many, many years. Okay? This is an image of an experiment done in mice where they took cells, they put a fluorescent dye in those cells. As a cell divides, the fluorescent dye gets diluted out. So after a couple cell multiplications, you no longer see the fluorescence. Okay? So what they did was they injected these cells into the portal circulation of mice, these cancer cells, these colon cancer cells. They go to the liver. And what they did was they go back into the liver and look at the liver many, many weeks later. What they found was that the cells they injected were sitting there. Their fluorescence was exactly the same as it had been at the day 0 at the initial experiment. They're just sitting there chilling. They're not doing anything. They're just hoping and waiting for some good stuff to come their way, and then they would expand, and they might be able to grow up. But this concept of dormancy is important. All right. We're gonna finish up this section on metastasis by giving a couple molecular examples of how metastasis can occur. Okay, this is just a little teasers, and then we'll just get into talking about cancer screening and a little bit of other stuff, and then you guys will be out of here. We'll get you out of here definitely before you're supposed to. So first example of metastasis I want to talk to you guys about is this cell adhesion property. To metastasize, cells must reduce their adhesiveness to each other, right? Or else they can't leave the primary tumor site. And one mechanism by which they do that is by decreasing the levels of this gene called E-cadherin, okay? Epithelial cadherin. This protein starts out in the plasma membrane, and what it does is it allows this cell here to attach to this other cell above it through adherens junctions, okay? Keeps them next to each other, okay? And E-cadherin is bound by this other protein in blue. See this protein in blue? That's called beta-catenin. Beta-catenin is linked to the actin cytoskeleton of the cell through this other gene called alpha-catenin, okay? So normally, there's this tether on the plasma membrane that is coupled to the actin cytoskeleton by this protein, beta-catenin, okay? As cancer cells start metastasizing, they reduce the levels of E-cadherin. When E-cadherin levels go, suddenly this blue protein loses its attachment to the cell next to it. So now the cell no longer has its adherens junction, and the cells are no longer contacting each other. During development, the cells are wired to know that once this contact is lost, then the cell should migrate, because developmentally, that's what happens to cells that lose their contacts. So how do they do that? This protein, E-cadherin, goes from the cytoplasm into the nucleus, binds the transcription factor, and turns on a gene. And that gene allows the cell to move. Okay? Pretty cool, right? So that's adhesion. The next molecular example I'm going to talk to you about is called invasion. Okay? So what do metastatic cells have to do? Imagine if right here, actually it looks like we're inside of a tumor. Imagine you got this ball, the auditorium, it's kind of a tumor. And everything outside that, so if you look up at the walls, in order for us to get out and metastasize, we have to chew through things, right? If this is the tissue, we don't have walkways, we have tissue around us, we have to chew through it. And how do metastatic cells do it? That's what they do. They chew through the tissue, okay? They have to chew through extracellular matrix, they have to chew through basement membrane. And how do they do it? What is all that stuff? What are those things, guys? Extracellular matrix, plasma membrane. What is it made up of mainly? Proteins, right? So we chew it up using proteinases, right? And that's what metastatic cells do. They increase levels of what are called metalloproteinases, which end up taking extracellular matrix and chewing it up. Okay? And but after they chew it up, it opens up space for them to move through that tissue. Okay? Before they do that, though, certain cells need molecular guides, and they need permissive signals, because they've learned that in order to move, they have to walk over a certain rug, for example, or else they can't walk. Or they need certain growth factors that activate their motors and make them move. So what do these clever metastatic cells do? Well, they just make those growth factors, and they make those permissive molecules that were only present during development in very confined regions. They just make it, and they put it in front of themselves, and then they walk over it. An example of such a protein is tenascin C. It's an extracellular protein that's expressed during development in your stem cells. And what these metastatic cells do is they make lots of it, and they put it in front of themselves. They chew up the extracellular matrix, they put this protein in front of them, and then they're able to migrate over it. Another factor is SOX4, a transcription factor that we identified, that also allows these cancer cells to move. So you guys might ask, well, how do you know that these things, these genes and these proteins allow these cells to move through tissue? What we can do in the lab is we can actually label cancer cells from patients with a fluorescent dye, and we can put it in these chambers. This is a chamber called the Boyden chamber, where you have two platforms, kind of like, it's kind of like you have a little hot tub here, and then it's got a base at the bottom where you put extracellular matrix proteins, and then you've got another compartment here, and you have holes here, small holes, and these cancer cells, in order to get to this other side, have to chew through that stuff, and they have to migrate through that stuff. And you can see that they do it, and they're very effective at doing it. And when you take away a gene like tenascin C or a gene such as SOX4 transcription factor, they can't do it as well. Okay? So the final example is what do cells do once they get into an organ, and how do they, break down these organs? And these, you guys have to imagine, if a cell that's supposed to really grow and used to the breast tissue suddenly lands in a certain other site, such as the brain or the bone, it's fairly foreign to it. And it's not very hospitable, so it can take a long time for it to degrade that tissue and degrade that organ. And so, as I told you, these cells are very clever. So one of the things that breast cancer cells do when they land in the bone is to say, as they start expanding, many of them die because they can't expand any further. But the rare cells within that population that have turned on a gene, for example, that can activate cells that chew up bone, that cell will have an advantage, okay? Because it's figured out a way to make more space for itself. It makes a growth factor that causes osteoclasts, osteo, bone, clasts, these are cells that break down the bone, to start chewing up the bone. And when it does that, it liberates growth factors and calcium. Those growth factors can actually make the cancer cell grow even faster. And it can allow the cancer cell to make more factors that activate the osteoclast. So this is a mechanism by which the metastatic cells can degrade the organ microenvironment. And the rare cells that have turned on that right gene can actually be successful at this process. And the reason that cells are able to do this extremely challenging task is because women who have breast cancers that are large, many cells will leave that primary tumor and get into the bloodstream and get to those sites. So there's a large number of cells that allow selection to occur. So our conclusions are neoplasms or cancers are heterogeneous. Heterogeneity and having a sufficient number of cells ensures that you get completion of metastasis in a fraction of patients. Metastasis is a stepwise process. So the properties that you gain, the phenotypes that you gain in the early stages can help with subsequent stages. So being able to leave a primary tumor and swim through a narrow endothelial barrier can help you swim through a narrow endothelial barrier in the lung, right? So stuff you learned in junior high school can help you in college and beyond. So that's sort of the way to think about it. Metastasis requires the function of many gene products acting in concert, okay? Maybe this is why we've had such a difficult time preventing metastasis, because we have to find ways of hitting multiple gene products, not just a single gene product, okay? And metastatic cells use genes used by normal, so one might say, maybe if 100 years ago you asked how do cancer cells spread, someone might say, well, these cancer cells are so clever that they can't come up with new proteins. They engineer all sorts of new proteins that do novel things that no one's ever seen before. The experiments tell us that these cells that metastasize are doing the same things other cells in our body do during development or during embryogenesis or during normal physiology. They're not supposed to do it, they're just doing it, okay? They're not creating they're not engineering novel proteins to do these things. They're just using the proteins that have evolved to do these things extremely effectively. They're just turning them on at the right times in the right places, okay? Okay, so everybody stand up, stretch, and then we'll finish this up with a little bit on cancer screening. Let's talk about screening, okay? So I would say that the most effective tools we have against cancer are really preventative tools, okay? As I've told you, cancers, from the pictures and everything, you guys have realized cancer is a bad thing. And it's complex and it's aggressive, and and obviously we're understanding the biology, and the next 100 or more years are going to be very exciting as we start unraveling this disease and as we try to get to the same point as we have with treatments for cardiovascular disease and infectious disease. However, I would say that the lesson here is that why wait for a car to break and then try to fix it? Just why not try to prevent it from breaking down through preventative measures? And that's what we're going to talk about here. So prevention, so for breast cancer, you guys we've already talked about mammography. And so this is how it works. Basically, this consists of a camera unit, and an X-ray beam, as well as a film plate. The breast is compressed, and basically images are obtained. These images are called mammograms. And what you see here is that in normal breast tissue is this dark color, and what you'll notice is that cancerous lesions typically have calcification, and that can be detected by the mammogram as these regions that show up more white relative to the surrounding regions. Once that is detected, one can then basically biopsy those lesions and find out if they're cancerous, and if they are, lumpectomy can be performed and to get a sense of what type of cancer you're dealing with. One can then find out whether there's spread to a lymph node, and then one can decide on the surgical management. And most of the time, depending on how far the spread is, sometimes surgery is all you need. Most of the time, you need certain types of chemotherapy. Those can be either agents that block hormones, such as the estrogen receptor. They can also be agents that are just simply chemotherapeutic that kill the cancer cells or prevent the cancer cells from metastasizing. So women over 40 years of age need to have mammograms. Clinical breast exams are recommended for women greater than 20 years of age or older every 3 years, okay? So these are recommendations that we're putting up by the American Cancer Society. And cervical cancer. So cervical cancer can be prevented. So we're not going to talk about the vaccine and stuff, but that's also an important issue that's of contemporary times. But one thing that's really important is Pap smears. Okay, so this is a straightforward process where a slight number of cells are obtained from the opening of the cervix, and one performs a staining for this region, and if you see abnormal cells, you can basically treat that region of the cervix, and cervical cancer is highly treatable and curable and preventable. Okay, so women need to have cervical cancer screening by 21 years. And just a disclaimer, you guys should all talk to your primary care physicians and your pediatricians, if some of you go to your pediatricians, about screening. And these are really important measures. And I can guarantee you that after we finish this, one of you is probably, at least one of you, or many of you, are probably going to, by telling others, such as family members and relatives, to get screening, at least one or maybe a handful of you are going to, your efforts are going to lead to the identification of some lesions that are precancerous that are going to be detected in friends and family. So highly encouraged, okay? So colon cancer, how do we prevent it? One thing that we do is we do colonoscopies, okay? So for those of you who don't know what a colonoscopy looks like, we use an endoscope. I don't do it, but the field gastroenterologists do it. They use an endoscope, and that consists of a long tube with a fiber optic probe and a camera at the end. It also has a tube that can both inflate the gastrointestinal tract, but it can also shoot in water to clear up certain regions so you can visualize it better. The idea behind colonoscopy is to detect lesions before they become cancerous, okay? And these are called polyps. But they can also detect cancers at early stage so that their likelihood for cure is much higher. And again, this is the reason for the big decline. This is the decline in colon cancer death rates and incidence. And here's what we have. So you guys can see this. I mean, this is normal villi in the ileum. This is a small intestine. You guys still go to water parks? I used to go to those when I was young. So this is what this reminds me of. So this is the orifice of the appendix. Okay, this is your appendix, and these are some lymphoid aggregates, right? You guys have heard of appendicitis, inflammation of the appendix. So this is normal appendix. In fact, we have an endoscope here. If anyone's interested, we could do a demonstration afterwards. Not so excited now, are you? All that enthusiasm. No one's jumping up and down. Okay, so this is the ileocecal valve. This is what separates the small intestine from the large intestine. And you guys see these folds. Do you know what these folds are called? So the scope is moving backwards. It's being pulled out of the, they're called haustra. It's being pulled out of the individual. We're almost there. So this is just some yellowish stuff that they're washing. And you can see there are no tumors or lesions. Everything's looking honky-dory. Okay. It's coming out. All right. End of story. Almost. All right. So men and women 50 years and older, all right? So parents, grandparents, okay? Go tell them to get this, okay? Parents can sometimes be stubborn. In my experience is men can be more stubborn than women when it comes to these screening measures. So you might have to incentivize them. So remember when your parents wanted you to do something, you gave them ice cream? Like, you guys tell them you'll clean your rooms if they do a colonoscopy. Okay, so this is what a precancerous lesion looks like. It looks like a polyp, right? And so the probe can go and pull this out and prevent it from becoming cancerous. Okay? All right, so this is just as important. We're going to talk about lifestyle measures and cancer. Okay? So this is really important. And so the most important thing, one of the most important things is avoiding tobacco. No one here smokes, so you guys will all be telling your friends and family about this. So tobacco avoidance is key, okay? Avoiding obesity, so maintaining a healthy weight, okay? We're not talking about people being super skinny, we're just talking about a healthy weight, all right? Obesity is a major risk factor for a number of cancers, okay? Limiting alcohol intake, okay? Alcoholism is linked to a number of cancers as well. So for you guys, it's really important to protect your skins, and most importantly, to avoid sunburns, okay? So this is really important. And if you do have moles or things that look abnormal on your skin, to have your primary care physician look at those lesions. To be physically active on a regular basis, okay? So physical inactivity, cancer loves physical inactivity. It's really important to stay physically active, to continue to exercise. You guys are going to go on to college. You might be studying all the time, not exercising, and you really should do your best to continue to be active, walk, etc. And eat your fruits and vegetables. You know, these are all things that your parents probably told you that you guys don't like to do, but let's do it. So now, what I'm going to do is I'm going to just briefly tell you about, just really quickly tell you about how I became a scientist, just a little bit. So going back to when I was your age, when I was in high school, I went to a high school summer program, and it seemed just like a lot of fun. And we went to the University of Utah, and we basically did summer research for 2 months, and had a great time. And I came back the following summer and did it again. And I went to college, and I was interested in being a doctor, so that was my goal. But the more, once I started my classes and was doing taking my exams and doing my coursework, I just couldn't help but stay out of the laboratory. I loved doing experiments, and it just became more and more of an addiction that I couldn't give up. And even when I ended up doing an MD Ph.D. program where I was both studying medicine and science, and even while I was in medical school, I just loved to go back into the lab and do experiments and do science, and just really had a fantastic time discovering things that other people had not seen. And so what ended up happening was I continued along the physician path, but I just realized more and more that this was really my passion. And I think what you guys should realize is that, as I realize, is that science plays a really important role in advancing society and creating for a better society. And it's not just a means to an end, such as it shouldn't necessarily just be a way for some individuals to go to medical school or to finish college, but I think for many of you, if you give it a try and you get into it, you'll see that it's an amazing part of life that I think you will all really enjoy, especially once you start actually doing it. Reading it can be something. I actually really enjoyed reading science books, but once you actually start doing it and you do more of it and you start figuring things out and coming up with questions and answers and more questions, it's just really fun. So with that, what I want to do is, I want to give out some of these chocolates. And so what I want to do is, so what we'll do now is we'll just let's just have 2 lines formed here. And whoever wants a chocolate, there's lots of it. Lots of chocolate here. So we're 10 minutes early. We're gonna end the talk here. Chocolates for anyone who wants it. I also have some Dayquil cough suppressant here if anybody wants it. Yeah. But thank you. Good luck. Happy New Year's.